AdAlta's lead candidate that you heard from Carol binds to CXCR4, which is a member of the GPCRs. And so what are these G protein coupled receptors? So they're large family, probably the largest in the genome, play a number of roles in normal physiology as well as in pathological states. And of the top 200 selling pharmaceuticals, about a quarter of them target a GPCR. And the total market value is pretty big, and you can see the breakdown there. So there's about 800 GPCRs, but 400 of those or so are olfactory GPCRs, and they're not really considered by most people as drug targets. But of the other 400, they are really considered to be drug targets, and only about 100 of them or so are actually targeted by current drugs. So there's a lot of opportunities, certainly in drug discovery. What they are very complicated membrane proteins. They span the membrane several times, and when an external signaling molecule comes along, it binds. There's a conformational change that leads to the binding of certain intracellular proteins, which then activate certain pathways. You can have things binding that will activate the pathway. You can have things binding to the GPCR that will inactivate the pathway. There's also this phenomenon called bias agonism and antagonism, and I'll speak a little bit more about that later. But what happens when those proteins bind is that the signaling pathways get activated, and they can be quite complicated. That again makes it quite challenging to pick a drug with the desired effects, but it also provides an opportunity maybe to very specifically design drugs to do the job. You've seen this picture before, Carol showed it. We're looking here particularly at the GPCRs. A lot of the GPCRs are like CXCR4. You can see that, and it's shown here in green, there's very little protein that actually sticks above the membrane. That's made it traditionally very hard to get antibodies, monoclonal antibodies. Terrific class of drugs, but they tend to bind very flat surfaces, and they don't have much in the way of epitopes and things to grab hold of. So traditional antibodies have been quite difficult to get. And the other thing is that the similarity, as Carol alluded to, between GPCRs means that sometimes when you get small molecules, there's off-target effects. It binds to not just the one you want it to bind to, it binds to other GPCRs and gives you unwanted effects. So the therapeutic opportunity that currently, the way these things are screened is by small molecules. And you have limited candidates coming from small molecule libraries, or toxins. A lot of natural toxins actually bind to ion channels, for example. Obviously they have problems in terms of developability. It's quite difficult to generate a toxin that will be specific and not have toxic side effects. Sometimes the small molecules, the traditional drugs, have got limited potency and then off-target effects that I mentioned, and then I've alluded to this inadequate access to the receptor. But there are high-value targets there, and they remain largely untapped, I would say, and sometimes very intractable. So the iBody solution that we would say would be, it's a small single domain. It has these long loops, and I'll talk a little bit more about that in a second. But what it allows you to do is to very selectively get binders to your particular target with limited off-target binding. They're developable, certainly more developable than, for example, toxins, and they're able to access these unusual clefts, active sites and ligand binding sites in proteins. Traditionally, the way these libraries have been screened is that small molecule libraries of sometimes hundreds of thousands of compounds, maybe even 1 million, are screened to identify a lead compound. You then need to do a lot of chemistry to try and get not just the small molecule that will bind, but also make sure it's got the right characteristics like solubility and various things like that. And often you don't get the best candidate, and it's very difficult to synthesize the optimal candidate. And often, sometimes in this area, you need knowledge of the structure to help you design these good small molecules. As I've said, antibodies are a great class of drug, but Often we develop antibodies by immunizing GPCRs into animals. That gets chopped up in antigen-presenting cells and delivered. And so I think it's very fair to say that in this area there are very few good quality functional antibodies against GPCRs. So the AdAlta library has got a large number of compounds, about 20 billion different I-bodies that we can screen very quickly. We don't need any understanding of the knowledge of the structure or things like that. We can also screen in a native format, so we can screen the GPCRs in a membrane. That's where it likes to be, and we think that keeps it very functional, and so the binders that we get out are actually, we believe, better. And it's because of this long loop that we can access those ligand binding sites. And again, we think they're very much more selective. So a little bit about CXCR4, Carol mentioned it. It's very important for maintaining stem cells in the bone marrow. And at a very controlled rate, you're able to release stem cells into the peripheral blood as needed. There is a drug on the market, AMD 3100, or called Mozobil, which is able to mobilize stem cells. It's approved by the FDA for a single use, because really what you don't want if you're administering this on a continual basis is continual release of stem cells into the peripheral blood. It was found a long time ago that HIV uses CXCR4 as a co-receptor. It also uses another GPCR, CCR5. And CXCR4 expression is very low normally in tissues, but it's upregulated in a lot of different cancers. And as Carol showed you, and the data that we've got with other collaborators, is that it's highly upregulated in lung eye pulmonary fibrosis, kidney, as Carol showed you, in the eye, and in scleroderma. And this is just an example of lung tissue, so very similar to the kidney picture that Carol showed, where no staining of the eye body in the lung, but then in the diseased IPF human tissue, you get massive upregulation and staining. So just a word about AMD. There is a small molecule, AMD 3100. I said it's approved for mobilization of stem cells. It's got a lot of side effects. It's very toxic in animals. It increases stem cells, which is not a good idea to continually do, unregulated. It can also cross the CNS. It can cross the blood-brain barrier, get into the brain, and it activates, well, it can bind to a lot of different GPCRs and ion channels in the brain. And I think that's what leads to a lot of the side effects. Also, Prolixafor has been known, so this molecule has been known to mobilize cancer stem cells, for example, in some patients with leukemia. So those are the problems really with the existing CXCR4 therapeutics. They all tend to mobilize stem cells. How do we get these I-bodies? We generated a scaffold, so this bit in red is the same for every single compound in our library. All 20 billion of them have got the same core structure. They just differ in these loops at the top, and that differs in amino acid and length. And so we can screen the library against a particular drug. We can then select one that will bind into exactly the right fit, and then we can take that and try and develop it as a drug. The way we do it is actually to put each of these little compounds onto a bacterial virus called a bacteriophage, and that allows us then to put the target on a solid substrate, throw in the whole library, and then in there's going to be one or two that might bind. They will bind, you wash the rest away, and then you look that off, you grow it up in bacteria, and you find out what it is. If biology was perfect, you'd only need to do this once, but of course you have to keep doing it, and we call it panning because it's a bit like panning for gold. In here there's a little golden nugget and there's a lot of rubbish, and as we keep panning we increase, and we get rid of the rubbish, and we find out what we want. And sometimes we'll get 1 or 2 binders, sometimes we'll get 10 or 20, and then we can adapt and modify those. But very quickly what we have to do is put them into in vitro culture, and so do they do anything? We also want to quickly go to human cells. Do they affect what we think they affect in human cells? And then quickly try and get into some in vivo model as well. This is the AdAlta I-body, a model of it docking into, so this is in red, docking into the CXCR4 with that long loop penetrating right into the cleft of the molecule. When we looked at our lead candidate and we screened it against 167 different GPCRs, we really only got any activity with CXCR4. There was very little activity against all those other GPCRs, so that tells us that it's extremely specific. So GPCRs, the challenge and the opportunity in GPCRs is really on this slide. What you have is external ligands binding to a GPCR, and they can be different ligands. So in a way, you can imagine this is a little microprocessor which is taking different signals, and then it's causing different pathways to be activated. It's probably better shown here. Sometimes you can get something that binds, it's called a balanced agonist. It binds and activates two different pathways, G protein pathway and beta-arrestin. So this is a bit oversimplified, but that's generally the case. You can also get something that will bind and maybe activate one pathway but not the other. And then depending on which disease state, maybe you want to activate this pathway, the beta-arrestin one, but not the G protein. So it should be possible to select ligands that do this sort of thing. And just as an example that maybe people are familiar with is the opioid example. So the opioid receptor is a G protein, a GPCR. And this was an article I just pinched from Science a few months ago. And again, this is a little bit oversimplified, but in general, when the classic opioid like morphine binds, you get pain relief, which is what you want, but the other pathway also activates respiratory depression, which is what happens when you get an overdose. And so the idea is to try, and a lot of companies now trying to do chemistry around this, to try and selectively activate the pain relief pathway and block the respiratory depression pathway, for example. And according to Brian Roth of University of North Carolina, a lot of groups are working on it. Somebody's going to get there, and somebody's going to get it right. We think we've got some evidence that AD114, so again, this is another example showing you how it's locking into the GPCR. We've got our lead candidate, and we've got two other sort of siblings. So these are very similar molecules to 114. They just differ by a few amino acids. And you can see when you put them onto the GPCR and you look to see what the important residues on the protein that it's binding to, they've got quite different footprints. So 114 binds one way, these ones bind slightly differently, and we think that that's what will activate different pathways. And some of the evidence for that is shown, well, actually, I summarized it on the previous. So I've summarized it. For example, we know that this 114 has got high beta-arrestin activity, and we know that this one, for example, has got low beta-arrestin, and this one falls somewhere in between. It's why we chose those three to look at. And you can really see it here, the different beta-arrestin pathways, when we add the different I bodies, and we did 10 of them here, we can see that they're all activating beta-arrestin at different levels. The cyclic AMP is less diverse. We also showed that it was able to block HIV in only those viruses that use CXCR4 as a co-receptor. And interestingly, it doesn't really affect the calcium influx, which is one of the pathways that's activated by CXCR4. So none of our I bodies, you have to add large amounts of I body to get any activity in this assay. Whereas AMD3100, the small molecule I mentioned before, activates calcium really beautifully. So there's a few orders of magnitude. So that says that our I bodies are actually quite different from AMD3100. Sorry, go ahead.
Just on that slide, what cells are we dealing with here?
These are transfected cells with CXCR4 on them. So they're not normal basal epithelial cells or something like that.
Have you tested this assay in different cell types? Meaning, because I know the GPRs change conformation
Sure.
based on the disease state, the cell
Yep.
type, the maze that they're in. So do you see this consistently?
You do see this in other cell types. It's not just in the transfected cell. I mean, we haven't gone into as much detail on this because we're looking at AD114 and what it does. But yeah, we never see calcium activation when we've looked at this, but we all see some sort of form of beta-arrestin. We've looked at it with a few different technologies as well, not just the Brett. And we know that these are all different. So it's really a question of how you select for which ones. At the moment, we're not clever enough to know that this one here will be good in this disease and that one there will be good in that disease. We don't know enough to be able to make those decisions at this point.
Why is it important to activate the beta-arrestin pathway?
Well, so in CXCR4 case, beta-arrestin pathway is one of the pathways that we believe is involved in fibrosis. It may be in another GPCR. It's the other way around, that you don't want to touch the beta-arrestin pathway.
So Derek, to your point, we gave Corey Hogerbone at Cedars-Sinai, who's an IPF expert, a panel of I-bodies, and 8114 had the best activity in human cells. So, and the other ones had very, very different activity. So, for example, one that Mick showed up there, 523, which has 2 or 3 different amino acids, doesn't have any activity at all in the IPF. So these different biased antagonists are having different profiles in different cell types, real cell types.
Did you take them into in vivo models and see differences there as well?
Yes. Yeah. Well, yeah. I mean, the only thing we haven't done. It's very difficult to measure beta-arrestin in a normal cell because these Brett assays, you need to label the beta-arrestin. So it's actually quite difficult to then say that it's solely because of the beta-arrestin. But we know that this is about the only thing that's really vastly different between those I-bodies. So given given this last bit of data here, that our I-body is very different to the drug that's been approved for stem cell mobilization, we asked the question, does our I-body, does our lead candidate mobilize stem cells from the bone marrow into the peripheral blood? And the answer is no. In a mouse model, in a humanized mouse model, and in non-human primates, we saw no evidence of stem cell mobilization. Even when we did the experiment in the presence of AMD 3100, which did mobilize the stem cells, our I-body didn't have any effect. So there's definitely a difference between our I-body and the way it binds to CXCR4 and the underlying pathways. And we believe that this then can be used for chronic therapy. And I think just to go on to that, this is some of the work that Corey Hogerbone did, which is when we looked at our I-body, he showed that it was able to block the migration of pulmonary fibroblasts from IPF patients, pulmonary fibrosis patients. So when you put our I-body into those cells, it stopped them from migrating. It slowed them down quite dramatically. But it had no effect on the migration of fibroblasts from normal lungs. And he said to us that that was really the sweet spot, because a lot of the other compounds he's measured in the system, they do one but not the other. So for example, nintedanib, it was great. It could block fibroblasts from IPF patients, but it also stopped the migration of normal fibroblasts. So, and again, we can't explain this at the molecular level yet, but the I-body is certainly doing something very different to all of these other, and certainly different to the other CXCR4 drug, but also different to the drugs that are on the market as well for fibrosis. So we have looked at the eye. We got an NHMRC development grant with Erica Fletcher at Melbourne Uni, and we're looking at eye fibrosis in the eye and wet AMD. And again, we've shown evidence in the liver and the kidney, as Carol showed you, and in the skin. And we've demonstrated quite broad anti-fibrotic effects in these models, and also some anti-inflammatory effects with animal models and human tissues. And we've done several non-human primate monkey trials, and we don't see any adverse effects. So this is just a little summary of the treatments. I'm not going to go through this. It's just that a lot of the binders to CXCR4 bind about the same affinity, but they've all got very different properties. And I think, you know, the main one we would say would be that the I-body does not mobilize stem cells, but all the other anti-CXCR4 drugs pretty much do.
How did you get it to have such a good half-life?
We've extended the half-life. So the half-life of the normal I-body, the naked I-body, would be somewhere in the region of 10-20 minutes. You put something on the end of it, which makes it larger, so it doesn't get cleared by the kidney, and it keeps it in the bloodstream longer.
German collaboration.
Yeah.
It's fair to say that's been a successful collaboration for
It certainly extended the half-life of this. Yeah, we certainly. I don't think we would have been able to do this with a naked eye body alone. There's a lot of other technologies you can use to extend the half-life, but yeah, this seems to be working.
What do you think? Sorry. Where have you got the data from?
Which data?
For the VMS antibody and Ablix's product.
So publications, yeah. So if you go to the publications, you'll see it. And the Ablinks is also published. They've shown clearly in non-human primates that it mobilizes stem cells as well. So that's all publicly available. I don't think we got anything proprietary there. We don't know. Most of the other groups are looking at cancer. Because it's highly upregulated, and so there's a lot of interest in looking at cancer. But there you've got to kill the cells. You don't necessarily have to do that when you're looking at fibrosis. You've got to stop whatever the fibrotic process is.
Would a naked eye body work in the eye?
Yes, it would. We've done the non-half-life extended eye body, and it works. Yeah, the eye's obviously quite privileged, keeps it there. It's a little depot, so it would work. But the passilated one works in the eye equally as well.
The plasma half-life, was that done in vivo or in vitro?
In vivo, in vivo in non-human primates. It's
Okay.
yeah.
So the absorption of the AD114 to its target is not an issue in terms of withdrawing it from
Well,
the plasma?
yeah. Well, that's a tough question. As I showed you in some of those tissues, there is no CXCR4 in a lot of the tissues unless it's in a disease state. So these are normal, naive, non-human primates. So I don't see it getting swept up into tissues. But there is CXCR4 molecules on the cells in the blood, and so they may be binding to that and then being released. It's clearly got a half-life, and it's clearly not lasting forever. So it's lasting a lot longer than when we put the naked eye body in.
For useful clinical impact, what kind of half-life would you be looking at?
Yeah, look, it depends again on the route of administration, but and you'd really like to be able to not inject this on a daily basis, but maybe that you'd have to on a daily. And again, it depends on the dose. So if the dose that you require for efficacy is low, then I think you can get away with injecting weekly. But if the dose is high, then you're going to have to do it daily or maybe even twice a day at that sort of half-life.
So with the collaboration, you're looking to further extend the half-life?
Yeah, well, we're always looking to see what the best technology out there is that would extend the half-life even more, But I think this one will be able to do what it needs to do, and the question is whether this is the one that goes into final trials.
We'll find out later this year.
Well, yeah,
So this would get you through the clinical?
yep, for sure.
Just on the dosing schedule quickly on the non-human primates, what was given to them, how often? You said no AEs, no DLTs?
DLTs, no. There was a transient increase in neutrophils for a few hours, but none of the liver enzymes, blood counts, platelet. Basically, they measured everything. There was no adverse effects that they could see.
And that was the same extended life molecule?
Yeah, with that half-life, and it was dosed multiple times. It was like dosed daily. I think it was for 7 days at 30 mg per kilogram, which is quite high. So it's not. We didn't just do it once. It's we think that with the clever use of assays, we should be able to get specific binders. Specific eye bodies that bind to ion channels and GPCRs that are able to activate the pathways that we would like to activate, and we've got a real expectation that we'd be able to get these biased ligands, either agonist or antagonist, for whatever GPCR we're interested in. And again, just to finish on the fact that there are a lot of GPCRs and ion channels involved in inflammation, in fibrosis. So there's certainly a lot of, it's a target-rich environment, and there's a lot of opportunity. And then that's just the summary, yes. if there's any other questions.
Yes, so it's a dumb question going back in time. How come CXCR4 hasn't been a prominent target for HIV drug research over the years?
Well, I think because it's the two, you need both co-receptors, and so I mean some of the other guys in the room would probably know better. But if you block CXCR4, you're only going to block the HIV that uses that co-receptor. They can still get in using another co-receptor. So that's where you would definitely need a combination of the drug that's already out there for CCR5 and for CXCR4. And then I think the other thing was that the phenomenal success of drugs and HIV really made this there's no point really in developing expensive therapies when the drugs were working.
You actually do have an approved CCR5 that you were on the market, and that was just commercially just marketed because of really other combination. I think there was a lot of focus on CXCR4 as well, and then there was another one, GC Scientific, but commercially it's just didn't really.
I mean, could we think about functional cure, for example?
Well, I think also if you go Sangama, for example, they were really using single finger to cut out CCR5, and that's how Sangama went to work, and they were wanting to convert it, they never did. So, but I think it's, if you look at the current HIV drugs, that there's not, you don't really need anything anymore.
I think when they were looking at CXCR4, they were using Mosaville, and so you use it at high doses, and it's cardiotoxic. So you've got a small molecule to a GPCR, and it binds to multiple GPCRs and multiple ion channels, and I think that's really why we're saying this antibody-like technology, like the iBody, can bind specifically and not have those off-target effects.
I think we saw that. I think that tainted the target in many ways, because even when we went to Corey Hogaboom and said, we've got this iBody to CXCR4, can you test it? He goes, no, I'm not testing it. I've already used Mosaville. It killed my mice. I'm not, and so we begged him. And then he wrote us a lovely letter saying, look, I was wrong, So I think there is a lot of that, sort of the target gets swept up in any of those sort of things that comes along. And we have to fight against that sometimes, but I think people are coming around now.
Mick, is there a lot of polymorphisms in CXCR4?
There's not a lot. There is one classic one, which is the WIMS, and that's a gain of function, of course. And there's a group working on that up at the Walter and Eliza Hall Institute. But that's about the only one that I'm familiar with.
In AD114, does it work in like a WIMS model? Is there
We haven't done.
I'm just thinking about the complexity here of trying to specifically target a specific changing receptor in a changing disease state in a different pathway.
Yeah.
What animal model do you have? I mean, as things moves forward, how do you select, even thinking further afield, phase 1, 2, how do you select your patients?
So.
Try and turn on and turn off. I mean,
Exclude the midwife, right?
that would be more obvious. But
Yeah,
if
yeah.
you're looking at fibrosis, and
Well,
all
I think
that,
I think what we're seeing now, and certainly in pulmonary fibrosis, and it looks like in the kidney as well, is that a lot. You saw Carol's data. A number of different diseases, they all lead to upregulation of CXCR4, and so we believe that the patients are going to have CXCR4. The question is, what levels do they all have the same levels? And there's a lot of different strategies. It's a very vexed question on how to select your patients. But you could look for people who don't respond, for example, to phenidone and nintedanib. And I was at a conference, the ATS in the States, and in some areas about 30% non-compliance. People just don't take it because of the side effects. So I think, you could look at that. In the other cases with, for example, if you were going to look at ophthalmology, you look at people that maybe don't respond to the AMD treatments and say, well, those guys need something. Maybe that's a patient problem. Is it sufficient?
do you think that's an easier model? I'm trying to get, is the strategy in going IPF and fibrosis, is it too difficult getting back to Bianca's target and knowing what you're hitting mechanism of action, right? It's very complex, but if you go to AMD, a thing close to my heart, because I Macugen, but beside the point, it's a much easier model. Is that something you're thinking about?
We've certainly been discussing that in the last few days in terms of weighing it up. IPF is a difficult area. We have no illusions about that. I think wet AMD is easier, but then I don't think the role of CXCR4 in the eye is as established as it is in a lot of the other fibrosis. So you need to be careful before you would say, we're not going to do IPF, we're going to do AMD. I mean, the data we're getting at the moment look very promising, and I think it will probably hold up. But do you make that commercial decision based on that data? I think that's something we need to discuss probably.
Mick, how is 114 delivered?
We've delivered this, so when we did the safety trials, we've done it subcutaneously and intravenously. And in the eye, we can the mouse models in the eye just directly intravitreally.
So in the commercially, how do you think it'll be delivered? Subcutaneous or?
I think subcutaneous will be the way to go to get those sort of doses. It could be, I mean, if there's no issues and we had enough drug that we need, it could be either IV or subcutaneous, depending on how you want the.
A daily dose?
It would be subcutaneous. I think is the easiest way to do that. Yep.
And have the PK/PD studies been done on sub-Q?
Yep. We've done them. We've got a really sensitive mass spec assay. We can detect the eye body in the blood. We've done the PK in sub-Q and in IV in non-human primates.
Can you discuss why you went with the German company's passivation technology rather than say another?
The good old standard. Yeah. I mean, we
It's just CSL have done something interesting with their
albumin. Yeah.
Albumin.
Yeah, I mean, we did look at a lot. We looked at a lot of those companies, and we spoke to a lot of them. I think XL Protein had certainly the scientific expertise. They also had, I think we had a really good relationship with them. They showed us some data; it looked great. So, and the thing was that you could make it as a single molecule. I mean, we did look at the albumin way back.
Yeah. I think we looked at PEG, so PEG, coupling PEG to the eye body, and the issues with that were it's got multiple steps in a manufacturing process. You purify the eye body and then you do a chemical coupling, and then you've got to purify it again. And the advantage of the XL proteins in that instance is it's a one-step process, but very much like a PEG in that it's recombinant. Another advantage against PEG is really that XL protein technology is low immunogenicity. PEG, there's a lot of antibodies at the moment that people have because PEG's in toothpaste and everything else medications, and so a lot of people, I think it was 10 or 15% of the general population have an anti-PEG antibody. And so when we spoke to pharma, they really advised against a PEG product. We've looked at serum albumin binders. We've looked at coupling the FC. We've looked at albumin. At the moment, we make 8114 PAS in E. coli, and we really wanted to stick to that PAS molecule because it's in an E. coli system. And so that's where we really came from. You look at PEG and the serum albumin peptides, and this PAS molecule really gave us, in a one-step manufacturing process, the ability to extend the half-life and manufacture in E. coli.
If you used a fragment, FC fragment, would you need to?
We'd have to express into
In mammalian cells. Yeah, which is doable. Yeah. I mean, and the technology is getting better and better for that. So.
The Excel guys published on their data is fantastic. We're talking orders of magnitude for half-life ones.
Yeah.
In vivo.
Yeah.
So the original eye body on its own has about a 20-minute half-life, like Mick mentioned, and this has extended it,
100-fold.
24 hours.
Are there any commercial XL proteins?
I think the most advanced would be just getting into the clinic now. There's another technology very similar to XL's in the US called Ammunix, and they have a product
Extend,
which is for I think it's one of the recombinant proteins,
it's like an insulin molecule. Yeah.
And that's in phase 2 or 3.
Are you still working on extending the half-life for future generation products?
I think the platform allows us to look at pretty much any half-life extension technology. And so, AdLinks as an example have their own nanobody which binds to FcRn, and they then recombinantly express that one product in yeast.
Yeah,
So
so
there's, I guess as a platform technology, we have an option to go with whatever half-life extension technology we're interested in. It makes the most sense.
yeah, we can put anything on the C-terminus of this, and it doesn't really affect the binding. So, and we have done, and we'll probably keep looking at better and better ways to do it. Mm.
How would that affect, so Carol sort of talked quite a lot about how exciting the technology was potentially going internally in cells. How
Yeah.
does, you know, changing the size of the molecule affect?
That's
Look, I
something
d-
we've probably got to look at.
Yeah, we'd have to look at it. Look, I think it's still a challenging thing to be fair. I think that's very challenging to get anything into the cell to bind to and activate or deactivate ion channels. So I think you need to look at a lot of carefully other technologies to aid it to get through. Even though the eye body is small, it's still a protein.